Hello, everyone. I'm Laura Almasy. Peter Kuchinoff, David Glahn, and I will be doing the solar workshop this afternoon. I would like to make this informal and interactive, so I will have, pauses for questions here and there, but please feel free to interrupt me and raise your hand and bring up questions as they arise that may, help clarify things as we go through, and it may be easier to ask questions while the slide you're thinking about is still on the screen, and so forth. So you may notice that I'm not John Blangiero. He had hoped to be here but had to cancel last week for professional reasons. So I'll be doing the first two parts of the workshop, playing the role of John Blangiero. This is the outline that's on the website. This is my reimagining of that. So I promise you that this covers all of the same topics that were promised, but I have sliced and diced them in slightly different ways. So I'm going to start with a little bit of the classical variance component genetic model theory that underlies the analyses that you can run in solar, and specifically how that model is used to estimate heritability, what covariates do in that model, association testing, and just a little bit about linkage. Then we'll take a break, and then I will introduce you to solar, tell you a little bit about where you get it, documentation, user support, and the sort of basic introduction to how to run the analyses that go with the theory I've described, how to get heritabilities, how to deal with covariates and association, and just a little bit about linkage. So I've put up there only a little bit about linkage because it's my impression that most people in this audience are working with data sets of unrelated individuals. Is that true? Hands up if you have families. Okay, so there are quite a few people who have families. Okay, twin. Hey, they're families. All right, there are more families than I had thought there might be. Those of you with families, how interested are you in linkage? How interested are you in association? Okay, all right. So I think we've got the balance right. More association, but the linkage is in there. So this is the key to the variance component models behind solar. We're imagining the world as a bell curve, essentially. Along the x-axis, we have trait values for some phenotype of interest, and along the y-axis, we have frequency. So few people have very low levels, few people have very high levels, most people are about average. We describe this distribution through two measurements, the mean and the variance. The key thing is that we're trying to explain the variance. If the variance were 0, everyone would have the same trait value. They'd all be at the mean. For, all of our traits of interest, that's not true. And the key is we want to understand why people are different from each other. So we want to understand the sources behind that variation. So conceptually, it's pretty simple. So when you convert that into formulas, we're taking the overall phenotypic variance in the trait that we've observed. We've observed that people vary. And on the right-hand side, we're trying to provide potential explanations for why people vary. And our first basic division is trying to break that phenotypic variance into a genetic component and a not genetic component. Since I'm a geneticist, I call my residual environment. But that's really a residual over there. The epidemiologists make their entire career out of my residual. Okay, but we don't want to stop there. Just being able to say there are genes that influence my trait of interest, some of my variation is due to genes, is an important first step, but it's only a first step. We can continue breaking these components down even further. We can break that genetic component into an additive component and a dominance component, a non-additive component. And so what is that dominance variance? It's kind of a slippery thing to deal with, and I think the easiest way to explain it to you is to have an imaginary locus that has two alleles, A and B. Three possible genotypes, AA, AB, and BB. And here I'm spreading those genotypes sort of across my x-axis of trait values. So the AA group has the lowest mean trait levels, AB is intermediate, and BB has the highest mean trait levels. I'm going to characterize my additive genetic variation in terms of the difference in mean trait values between the two homozygotes. I'm taking half that difference and calling it A. So the BBs are plus A and the AAs are minus A. If this heterozygote deviates from the midpoint, the extent to which they deviate from that midpoint between the two homozygotes is D. And that is going to play into our dominance component. So if the heterozygote is halfway between the two homozygotes, that is, if the mean trait values of the heterozygotes as a group are halfway between the mean trait values of the two homozygotes as groups, then we've got essentially a dose-response type effect of the B allele. Each dose of the B allele is raising the mean trait values by the same amount. That's a purely additive model. If that's not true, if the first B allele raises your mean trait values a little bit, and the second B allele raises your mean trait values a lot, then you've got dominance components. So we can, for any locus that we know these things about, where we've got these mean trait values for the three genotypes, and we know the frequencies of those genotypes, we can directly calculate the additive genetic variance due to that locus. It's a function of that displacement between the means, and P and Q here are the allele frequencies, the frequency of the A and B alleles. So the thing is, if that D is 0, this term drops out over here, and the additive genetic variance is just 2pqa squared. If that D is 0, this entire dominance variance zeroes out. We've talked a lot about epistasis in the last couple days, so we can also add an epistasis term onto our potential sources of genetic variance. You get epistasis when A or D is a function of your genotypes at another locus. When the mean trait values for these AA homozygotes depend on whether they do or don't have the risk allele at some other locus. So it's pretty simple, conceptually. We can also do the same thing with that environmental component. We can break that down into some sort of shared environment and some sort of unique environment. But again, this unique environment out here, really a residual. Okay, so heritability has come up a couple times during the last few days. Heritability is the proportion of the phenotypic variance in a trait that's attributable, in the cases when I use it, to the additive effects of genes. So broad sense heritability is that total genetic variance component over the total phenotypic variance. What proportion of our phenotypic variance is attributable to our genetic component? The heritabilities I'll show you are additive genetic narrow sense heritability, which are just that additive component over the total. It's been brought up before in the last 2 days that this is a ratio. I can change my heritability by changing the genetic signal or by changing the overall phenotypic variance. If I increase, if I conduct a study and I've got a great heritability for my trait, it's 80%. It's got a very strong genetic signal. And I send my protocol over to my friend in Sweden, and he tries to do the same study. If I haven't quite explained things well enough, and he is not able to measure the trait as accurately as I have, he might get a heritability of 0.4 or 0.5 because the extra measurement error is increasing the phenotypic variance down here. So you can change the heritability not just by changing the genetic component, but by changing the measurement error or the environmental factors as well. So heritability is really a snapshot having to do with a particular sample measured in a particular way at a particular time in a particular place. It's not a Platonic truth that is the same for that trait everywhere or any time or any population or so forth. The other key thing about heritability is that it's an aggregate. It's an aggregate of the additive genetic effects of an unknown number of genes. So, I showed you back here, one gene. I've got the additive genetic effects for all the genes influencing the trait, and I've concatenated them together. How many genes? We don't know, and we can't tell from this. So higher heritabilities are better, but they don't guarantee you anything. Height has one of the highest heritabilities. IQ has one of the highest heritabilities. But both of those have not proven easy to identify genes for, because underlying those heritabilities are dozens, hundreds of genes. So the effect sizes of any of those genes could be very small, although in aggregate they are large. So given that I don't actually know the displacements between the trait means and the allele frequencies of all the loci I'm looking for, how do I calculate this genetic component if I don't have those numbers that I showed you before? And the way we do it is through a very simple model where we're modeling each person's phenotype as a function of the overall mean in the sample, measured covariates that we're allowing for differences in the trait mean, and then the additive genetic and environmental effects, which we're going to model through random effects on the trait variance. So we have fixed effects on the trait mean and random effects on the variance. And here is the variance part of the model. So on the left, this omega is the covariance among family members. For heritability, you have to have family members. Covariance is related to correlation. So you can think of this bit on the left as being a matrix of how similar and dissimilar all the relatives in a pedigree are to each other. The bit on the right are our friends, the genetic and environmental variance again. And then in front of each of those variance components is a structuring matrix that describes what the correlations among family members should look like if they're due to that component. If there's an additive genetic component to the variation, then the correlations among family members should follow Mendelian rules of inheritance, essentially. So this 2 phi here is the kinship, two times the kinship among individuals. You can write that down just based on the pedigree structure. You don't need any genotypes, you don't need any markers. Just based on the pedigree structure, you can predict what the correlations among relatives should look like if they're due to that additive genetic component. This kinship, this 2 times the kinship, 2 phi over here, is also the prediction of, yes?
Sorry to find
yeah, no.
The matrix omega, as it is sometimes particular in the particular data set, could it be that there's some estimation problem? When you've got two...
That is, well, I'm not sure what question you're asking there. So that is calculated from your observed data that you have measured about your phenotype.
Right, but it could be a big matrix.
It could be big,
and
yes.
Could have many parameters.
Yes, so...
So is there always enough zeros in that matrix that you would have that much of trouble with estimating, or that's the cases where you have?
It can be computationally intensive to deal with, but we have successfully, actually the biggest I have ever dealt with in this is one of the primate centers wanted to look at heritability of birth weight in their macaques, I think it was. And they had data, longitudinal data over the sort of tenure of their primate center on 20,000 animals born in the center that essentially coalesced into one pedigree. And we were able to successfully analyze that. So I think for practical purposes, The bigger the pedigree gets, the more computationally intensive it is, but it's still doable. We have not really, we have talked to some of the people with the really big human pedigrees. The biggest human pedigree we have is about 2,000 people. But we have talked to and to some extent worked with the people who are studying various things in the Amish. And there, it gets computationally intensive enough, and you have to go back far enough to connect everyone that it becomes problematic. So there are cases where it can become problematic, where you have, 10, 12, 14, 20 generations of unmeasured individuals who are needed to connect everyone. Then the computationally intensive gets to the point where it's not worth the diminishing returns of connecting everyone. Yes?
Sorry for a really genetics naive question, but how does that take into account common environmental influences, or can you ignore them?
An excellent question. We'll get to those in a couple slides. Foreshadowing. Okay, so this 2 times the kinship, there we go. This 2 times the kinship here that predicts the additive genetic component. That is the amount of DNA we would expect that type of relative pair to share on average. So MZ twin pairs are identical for their DNA, right? You have half of the DNA from either of your parents. They each gave you one of their 2 chromosomes. You have, on average, you share half your DNA with your siblings. Any particular pair of siblings, maybe more or less than that, but siblings as a group share half, and so forth and so on. And so basically what this is predicting is a very structured fall off in the correlations among relatives as you get to more and more distant relatives, if there's an additive genetic component to the variation in trait values. So reasonably simple, but you do need relatives who are all measured in your study to be able to make that prediction and comparison. So we can do some hypothesis testing with our heritabilities. Basically, we compare a model where the additive genetic component is estimated to a model where that additive genetic component is forced to 0. And we do a likelihood ratio test. Twice the difference in natural log likelihoods between the 2 models is distributed as a mixture of chi-square distributions. It's a mixture because in the nested model, we're forcing that component to a boundary. 0 is a boundary. Your variance can't go negative. Yep. So very simple 1 degree of freedom test is the additive genetic variance greater than 0. And then we can parameterize that in terms of the heritability. So heritabilities vary between 0 and 1. Higher indicates stronger additive genetic variation component. These are heritabilities for some random biomedically related traits from the, mostly from the San Antonio family study. Thrombosis is from another study I'm involved with, the GATE study. Alcoholism is from COGA. So, as was noted, we're assuming now that the only source of correlation among family members is genetic. And we know that that's not true. We know that families share environment as well as genes. The solution to most of those sorts of limitations with this model is to add a component to the model. So we can add in this C for common or shared environment. We just expand our partitioning of the phenotypic variance to include that common environment component as well as the additive genetic and the unique environment, really a residual. And we can come up with a proportion of the variance due to that common environment component the same way we do the heritability. The key thing is that to add a component to the model, you need a structuring matrix predicting what the correlations among individuals are going to look like if they're due to that component. As long as you can come up with that, you can add pretty much anything you want to the model. So, you'll see H as the structuring matrix on my slide here, because something we commonly do in our studies of heart disease is to use household as an environment, essentially, as a proxy for diet of people who are eating dinner together every night, probably a proxy for how much television they watch. You know, various things you can think of are correlated among members of a household. Basically what happens is that we number each household from 1 to whatever, and then we create this giant matrix by taking each pair of individuals and asking, are they in the same household, yes or no, 0 or 1? Do they share this? Do they not share it? It doesn't have to be household. We could do the same sort of thing for, we have some studies looking at genetic susceptibility to infectious diseases, where we might ask whether the individuals get their water from the same well in some of our more remote locations. If you are interested in developmental traits and childhood behaviors and that sort of thing, you might look at elementary school. Do kids in one school versus another share certain things? The thing that is automated in Solar will take that numbered category and make you a matrix that says share, don't share. But if you make your own matrix, you can put in shades of gray. There's no conceptual reason that this has to be 0, 1. It can certainly be anything in between. So for that study of infectious diseases, we have also done things like get the GPS coordinates of each household and then create a distance matrix among them. Question?
So can you break that down if you wanted to have H1, C1, H2, C2?
Yes. Yeah, you can. The thing is, you want to use these for environments that are categorical and numerous. If you have something that you can measure quantitatively, like if you can, I don't know, put a little gadget in everybody's house and measure the level of some pollutant in their house, that would be better. And you would want to treat that as one of the, as one of the measured covariates. So if you can actually measure it quantitatively, you want to deal with it as a covariate. If it's categorical, but it only has 2 categories, like smoker, non-smoker, then you can easily deal with that as a covariate. It gets difficult to deal with things as covariates when they're categorical and have a bunch of categories. You know, if you've got 9, you know, my example of the 9 wells in the village that people might be getting their water from, we could deal with that by creating a sort of dummy covariate for each of the wells. Do you get your water from this well? Yes, no, and so forth. Once you get much above that, then that's where you want to use that, that matrix sort of model, because you don't want to be creating thousands of dummy covariates and carrying them around. Okay, one of the assumptions of variance component analysis is that our trait is normally distributed. You remember, we're viewing the world as a bell curve. What happens if that's not true? Because we all know that the world is imperfect and few things are sort of the way the models conceptualize them. The key factor that, the key sort of non-normality in terms of the variance component models is kurtosis. So you can think of a couple ways that things don't look like bell curves. If they're off to one side, if they're skewed, the mean is not in the center. That's not so bad. The model is pretty robust to that. Kurtosis is when you have too many or too few traits out in the tails, essentially. And it's specifically leptokurtosis, fat tails, that are the problem for variance component-based genetic analyses. Those fat tails out there can inflate the false positive rate, essentially, for a variety of variance component-based tests. So the good news is that there are easy solutions. You just have to be aware of it as an issue, pay attention for it, and it's easily fixed if you find it. So here's the simplest solution, and the one we use most frequently, is basically normalizing the trait values. Just taking whatever you've got observed, and the blue lines here are real observed trait values for something, and normalizing it so that it basically conforms to that vaguely bell curve shaped thing. This is data from the San Antonio family study again. On the left is waist circumference. You can see we have some people, we have a very long right tail on this. We have some people who are quite obese and have very large waist circumference in the sample. On the right are leptin levels. Leptin is related to obesity, essentially. And again, we've got some reasonably long tails, and it's skewed. And there's a strong sexual dimorphism for leptin as well, which complicates things. But essentially, simple transformations can make these into our normal shaped curves. Now, there's trade-offs to imposing this bell curve worldview on our trait. On the one hand, it's protecting us against inflation of false positives. On the other hand, to the extent that there are some true signals out here in the tails, and there probably are, we may be decreasing our power to find them by normalizing the traits. So this individual is pretty extreme relative to the mean. Once I've normalized, they're less extreme relative to the mean. So it's a trade-off. In return for protecting against the false positives and allowing us to have a more robust analysis of the entire sample as a whole, we're trading off some power to find some of the things that might be out in the tail. Yes?
So in one of our studies, we actually looked at the blood pressure extremes.
Mm-hmm.
Therefore, the peak actually is 2 twin peaks.
Yeah.
For normalization. So when we do attempt to normalize that, do you suggest we combine the two extremes and then normalize them all, or actually normalize them individually?
Yeah, that's a really tough situation. It's difficult to do quantitative trait analyses in case control samples, because if you imagine the world originally was a bell curve and you've taken the upper tails of each and cut out the middle, then it really doesn't conform to the assumptions of this sort of analysis again, and it's hard to analyze it quantitatively with this sort of method. There are other approaches out there that I think would be more suited to analyzing that kind of data that don't have to make these same assumptions. I am the wrong person to steer you toward them because I tend to use the bell curve sort of data and thus don't have a lot of experience with those methods. Other people in the audience might be able to make specific recommendations, though. Or you can continue to analyze it as a yes-no sample, but I think you are losing power that way by not using the quantitative trait values that you have. But I don't think this would be the optimal method for analyzing the quantitative trait values from that kind of sample. Yes?
So isn't the assumption actually that the residuals are normal?
Yes.
So a transformation like this doesn't necessarily ensure normality of the residual.
Yes. So, for example, you may want to apply your covariates before doing the normalization. Yeah, that's a good point. And you know, particularly in the case of leptin, as we were saying, there's a strong sexual dimorphism, and accounting for sex is going to drastically change the shape of things there. Yeah, good point.
Do you use any criteria for rejecting your approach? In other words, for saying that data normalization transform is simply not sufficient to proceed with the analysis?
Two sorts of criteria. So, yes, I think there are two things you can look at to help you decide whether this is doing you any favors and whether it solves the problem sufficiently. So you can look again at the kurtosis of the distribution, and you know, if this has done what it's supposed to do, that should have gone away. And that solves the false positive problem. but something that you can look at that I think is a good use of heritability. It's come up a couple times throughout the couple days. What does heritability mean, and can you compare heritability between different regions of the brain and so forth? I think a legitimate heritability comparison that you can make is what happens to the heritability of my trait values when I do certain things to them. So if the heritability just goes away after this transformation, then it seems to be destroying the sources of your genetic signal. And continuing with your analyses with this transformed trait may not be getting you where you want to get to in that case. And I think other legitimate things that you can do with that are to look at those covariates again and say, am I doing myself favors with these covariates or not? Are they increasing my genetic signal, essentially? So I think looking at the heritability, whether your data manipulations are increasing your genetic signal as measured by the heritability, is a valid thing to do if your final endpoint is your association tests. So doing things that maximize your heritability should not bias the eventual outcome of your association tests, for example. Okay, so what about those covariates? We've got our measured covariates here. They're going to be modeled as effects on the trait mean, allowing the trait mean to differ in males and females, to allowing the mean trait values to change with age, be different in smokers and non-smokers, et cetera, and so forth. So if we imagine the total phenotypic variance now as a circle, and some part of that circle is taken up by the effects of genes. So G4 has the biggest effect, G1's a little lower, 2, 3, and 5 are a little lower. Our heritability is the sum of those genetic effects over the total variance. Covariates can effectively absorb some of that total variance so that we are no longer worrying about explaining it. We can put it in the explained column and have it accounted for, essentially. We can magnify our apparent genetic signal by accounting for some of those non-genetic effects, non-genetic variance, and taking that out of our equation. So now G1 is a larger portion of the revised total after we've dealt with age and sex. So the covariates can, even if you are not interested in the covariates as an endpoint in themselves, having them and accounting for them can effectively increase your genetic signals and increase your power to find the genes you're interested in. But you have to choose your covariates intelligently. Oh, before I tell you about choosing covariates intelligently, you can say, I know for valid a priori reasons what covariates are important for my trait, and I will choose them and include them. Or you can test them in your sample and say, I want to screen my covariates and keep the ones that appear to be important in my sample, or see if I can identify new covariates of importance in my sample. And the screening of the covariates basically, again, is a likelihood ratio test where you're comparing a model where a regression coefficient for that covariate is estimated to a model where it's fixed to 0. And in this case, it's distributed as a chi-square with 1 degree of freedom, because that 0 is not a boundary in this case. The beta for the covariate can be positive, it can be negative. We can have our trait values increasing with age, we can have them decreasing with age. Both of those are valid. So again, a simple test for is my effect different from 0? But back to those covariates. There's a danger in the covariates. You can choose a covariate that is itself genetically influenced. And when you do that, it will absorb some of the variance, but it may be absorbing genetic variance as well as environmental variance. So let's say I include BMI as a covariate, and genes 1, 2, and 5 influence BMI, and in fact influence my trait I'm interested in via BMI. Including BMI as a covariate, I potentially increase my power to find 3 or 4, but I've decreased or eliminated my power to find 1, 2, or 5. So if the thing you want to include as a covariate could itself be the subject of genetic analysis, you want to think long and hard about the question you're setting up and by including that as a covariate, essentially. So now I'm looking for loci that influence my trait independent of BMI. And maybe that's the question I want to ask, but you should do it in a deliberate way. Yes?
But here is sex as genetics, right? So
Yeah.
something you wouldn't include at all, ever?
I always include sex. So clearly I'm disobeying my own maxim there. Yes, sex is genetic. It's based on your chromosomes. It's XXXY, right? Somehow I consider it demographic, though. So I can't, yeah.
What about age in a certain situation? For example, Alzheimer's disease. There is factor for Alzheimer's disease age. As age, we know, is expression changes over time, which depends on the composition of your genetic background. We need to put age into your equation in terms of when you are studying Alzheimer's disease.
Yes, but let me come back to that and go back to the sex question. I don't have a good answer for that. I think most of the time, I think most of the signals we're typically looking for are not on sex chromosomes, or we're discounting the possibility of looking for them on sex chromosomes. You notice that it's very rare that genetic analyses of complex traits, whether linkage, GWAS, anything, possibly a few of them deal with the X chromosome. Of those that report results for the X chromosome, few of them are using the sort of models that account for the fact that females have 2 and males have 1, and almost nothing ever is looking for variation on the Y chromosome, except for traits that we know to be not just sexually dimorphic, but sex-limited. Then I think the question becomes different. But yeah, that's a good point that I should revise my language in talking about this, because sex certainly is genetic. On the question of age, I think in the specific example you raise, age is important to include as a covariate there, because exactly for the reasons you state, we know that risk changes with age, that
Whatever age is a risk factor for Alzheimer's disease. But if you see age itself is the changes of the expression profile, you know, over time, aging. And what is the changes of the expression profile is depends on the genetic background.
Yep.
So that's why my question, because I look
Yeah.
when I put age, actually I'm taking out so much, in my view, so much genetic variance from my phenotypic variation and variance. What is it?
So maybe the question you're asking is really about genotype by age interaction. So the effects of the genetic variance get stronger with age in this case.
Yes.
When you're young, having a bad genotype is very unlikely to put you over the threshold. When you're older, having the risk allele is much worse for you. That I would categorize as genotype by age interaction. And so you remember we're conceptualizing our phenotype model in terms of the mean and the variance. Including something as a covariate allows for an effect on the mean. The sort of model you're describing is a case where the variance attributable to the locus, that sigma squared A attributable to the locus, changes with age, where the A and D, you remember from our genetic variance equation from our pretend locus that had genotypes A and B, where A is a function of age. A gets stronger with age. So the way to incorporate that specific kind of effect is either, well, okay. These are models I don't have in there, but they're interesting ones and relevant ones for this group. You can make this additive genetic variance a function of age as well. You can allow for a genotype by age interaction. It takes a little bit of sophistication. It's a little more difficult to do, particularly with a quantitative trait like age. With something like categorical like smoking, it's easy. I can model one additive genetic variance for smokers, one additive genetic variance for non-smokers, and see whether, for example, keeping those two phi's constant, see whether the correlations among SIBs who are concordant for smoking differ from the correlations among SIBs who are discordant from smoking. That's the sort of thing you're talking about. It's more difficult to do that for a quantitative covariate like age. A lot of the traditional models for that require you to specify a shape, to say, I'm going to let my additive genetic variance change with age, and test whether it changes or whether it's flat with age. But you have to provide a model for that change with age. Is it linear? Is it nonlinear? That sort of thing. Vince Diego at our place has been working on some new models for gene-environment interaction with quantitative environments that use basically a distance matrix for the quantitative environment and incorporate that into this. And that you don't necessarily have to specify a shape for that. Peter?
Yes, I was just about to mention that actually this functionality is a part of solver.
Yeah, you can do it.
You can model with a function called
G by E.
It's not very powerful analysis, but it actually gives you a probability of the gene by age interaction. You need a lot of subjects to really get good probability, but you can do it.
Thank you.
Okay, so the dangers of including BMI or something like that as a covariate. You're asking a different question, and it's a focal question that maybe you do want to ask, but you have to think intelligently about that. I sometimes have difficulty, particularly in heart disease-related studies, where everyone knows that, let's see, our worst case, we were looking at left ventricular mass. And everyone knows that LV mass is correlated with a number of other important things like blood pressure and this and that and the other that the clinicians knew were clinically important. But from my perspective, I was afraid that the correlations between those two traits might be due to pleiotropy. And if the correlations between those traits are due to pleiotropy, by including that as a covariate, I'm potentially decreasing my power to find those pleiotropic loci. That's really the issue. But there may be cases where I say, I'm not interested in just any locus that influences my trait. I'm interested in loci that are obesity independent. And that is my specific hypothesis. Then this is helping me. Yes?
You have a vectorized trait, so you actually profile it as a
Yes. Yes, absolutely. That's a great comment. So even better is to actually jointly analyze my phenotype and BMI and exploit the correlation between them. And that can help increase my power to find those three, because I've got a more specific hypothesis. I'm looking for the thing that influences both of these. I don't discuss the bivariate methods here, but I think Peter, who is going to give some examples of imaging-specific analyses, does have some of the bivariate stuff in his. So he will talk a little bit about how you do that sort of bivariate, multivariate model. Okay, so the standard association analysis in the variance component model is essentially treating genotype as a covariate, really. It's testing whether the trait mean differs by genotype, which is exactly the sort of thing we described earlier when we were talking about those A's and D's. The advantage of this model over just doing a regression in your standard stats package is that if you've got families, it's taking into account the non-independence between family members. So we can define a QTN-specific heritability in the same way, the proportion of variance due to my locus. And here by QTN I mean quantitative trait nucleotide. Hopefully in the end game that I'm trying to get to, I've got in my hands a genotyped true functional variant that I would like to test whether or how the mean trait values vary by. So we're trying to effectively, by including genotype as a covariate, we're trying to estimate these A's and D's. And that QTN variance, again, is 2p, 1 minus p is q, so I'm changing the terminology on you, but it's the same equation, 2pq a squared. That 2pq, or 2p 1 minus p, or h up here, is the heterozygosity of the marker. So there's a key concept behind here. My QTN variance and QTN-specific heritability is a composite of two things. It's influenced by the extent to which the mean trait values are displaced. You can think of that as an effect size in an individual. So for an individual who has the B allele, how much higher are their cholesterol levels? And this 2p, 1 minus p, the heterozygosity, the allele frequencies, takes it up to an effect level on the population. So it may be that I have a variant that causes a gigantic increase in cholesterol levels in each person who carries it, but it's extraordinarily rare. So the QTN variance accounted for, the variance in the population that it accounts for, could be small. Or it could be that I have something that's very common, but it causes a tiny increase, a tiny difference in the mean trait values. Those are the things we're finding now by GWAS. And again, the effect sizes on the population level are going to be small. So a word here about those small effect sizes on the population level. Those are still valuable, particularly in phenotypes where we are still trying to understand the underlying biology. Having one of those small things that are present only in a few individuals can be an absolute breakthrough in understanding the pathways, the systems, the biology that's important for our phenotype. So familial hypercholesterolemia is rare. There are a few individuals who carry these giant whopping, essentially monogenic genes that cause giant perturbations of the cholesterol levels. But studying those families has taught us a lot about the mechanisms underlying heart disease. So just because something has a small effect size in the ways we measure it doesn't mean it isn't important scientifically. So how do we use that genotype as a covariate practically in our analyses? And essentially, we take that genotype, AA, big A, little a, AB, whatever, and we assign numbers to each of those genotypes. If you have a big A, I'm giving you a 0, big A, 1, little a, 2. This is essentially the number of minor alleles that someone carries, 0, 1, or 2. Yes?
Do you ever model 4 different genotypes, meaning that the heterozygous group can also have either a maternal or paternal big A or small a in this case?
Me personally, no. Could you? Yes. Yeah, so this is almost infinitely customizable. Any model you can describe of gene action, if you can figure out how to make the covariates for it, you can test it, essentially. So I'm saying under an additive model here because I've forced the heterozygote mean essentially to halfway between the two homozygotes. But if I don't like my additive model and I want to include that dominance component, I can make another covariate here that's 0, 1, 0, and that essentially allows the mean of the heterozygotes to move around from that midpoint. So I've complicated my hypothesis testing in that case. I then have to decide whether I'm testing the additive component, the dominance component, or a 2 degree of freedom joint test of both. which is, increasing degrees of freedom, potentially decreasing power, unless you need that in there, and then increasing power. So it gets sort of tricky. But you can certainly do parent-specific effects, parent of origin kinds of effects as well. Easily manipulating it by number of little a alleles inherited from the mother, or number of little a alleles inherited from the father. It's really easy to customize these kinds of models. You have to have once you understand them deeply, then they're really easy to customize just by changing this covariate that you're giving everyone, or by creating a new structuring matrix. You can do almost anything. Okay, so power comes up in grant applications and in discussing complaints about imaging genetic studies having too small sample sizes and so forth. So let's talk a little bit about power. The power for association studies is a function of the sample size, family configuration, that QTN-specific variance, and if you don't have the functional genotype in your hand, the linkage disequilibrium between that functional genotype and what you do have in hand. So the effective marker-specific heritability that we're operating on for our power is the effect size of the functional variant times the R-squared measure of disequilibrium between the true functional variant and what we've got genotyped. So GWAS panels were optimized to maximize that R-squared between what's on the panel and common variance. The R-squared between what's on the panel and less common variance is worse. So it's possible to me at least, that some of the weak GWAS signals we're seeing are really potentially poor LD between rarer loci of stronger effect than we're estimating based on common SNPs. So some of that disappointing GWAS signal could be due to having poorly measured rare variation, essentially. Of course, once we get down to the whole genome sequence, this question to some extent disappears. We no longer have to worry about the LD between our functional variant and what we've got genotyped, because we've got everything genotyped. Then the problem turns on its head and becomes different. Then the problem becomes, of the 15 markers that are all giving me p-values of 10 to the minus 27, how do I figure out, that are all in LD with each other, how do I figure out which one of those is functional and which other ones are just in LD with that functional one and therefore giving me good p-values? And it can be difficult to impossible to do that statistically, and that's where the biological knowledge and biological experiments come back into it again. I say family configuration in here. So family members are correlated with each other. We exploit that in our studies, but it also is a slight disadvantage for, well, it's both an advantage and a disadvantage for these sorts of association analyses. It's an advantage because I can get more copies of those rare variants, but they're not independent copies of those rare variants, essentially. So if I have 6 people in one family, they don't give me quite as much power for my standard association study as 6 independent people. The effective N that I have with related individuals is lower than my actual N. Okay, so let me give you an example of this simple measured genotype association analysis. You remember, we're testing whether the mean trait values vary by genotype. Extremely simple. This is a functional variant in the clotting factor 12 gene, and clotting factor 12 levels vary strongly by genotype here. So individuals with C alleles have much higher average levels of clotting factor 12. Individuals with heterozygotes are more or less intermediate, and the TTs have very low levels. Easy, simple. So we're testing whether the mean trait values differ by genotype or whether our regression line is really flat and there's no difference here between genotype. For people who think in pictures, this is another example from the same study of clotting factors, but it's prothrombin levels now. So we've got our 3 genotypes, the common homozygote, the heterozygotes, and only 4 of the rare homozygote. And these lines here are drawn in at the mean for each group, and we're attempting to draw a regression line through that and say, is that different from 0? Are the means really the same for these groups, or do they differ from each other? Since I'm using an additive genetic model and attempting to draw one line here, I'm sort of forcing this middle group to be halfway between the others. And as it turns out, that's not a bad fit. I think that people's beliefs about non-additive effects are strongly influenced by the type of data they're working with. I tend not to use models of epistasis and non-additive effects because in the things that I work with, I see a lot of pictures like this and a lot of numbers like that, where that simple additive model is a pretty good fit to the data I have. So I tend not to have gone further into those sorts of models. But it is perfectly possible to do that, and not only to let your regression coefficients here be a function of age, but to also let them be a function of genotype at some other locus. So you can directly model those sort of epistatic effects here. Again, you take a power hit when modeling those sorts of things. Another point here, we're testing differences in the means, but look at the spreads here. Look at the variance within each of these genotype groups. It's big. I mean, these groups are overlapping. As a group, the mean trait values in heterozygotes are higher than these homozygotes, but there's no heterozygote here who is more extreme than the most extreme homozygote. What this means to me is that there is still unexplained variation within these groups. In the graph I'm showing you here, we're not accounting for things like age, sex, and smoking that we know about. But even if we account for those, there's still variation here. These are complex traits. We expect them to be influenced by many sorts of things. Okay, just a little bit about linkage analysis. Linkage analysis, unlike association, we're dealing with as a variance component, as an effect on the trait variance rather than the trait mean. So we have a new variance component here, our QTL-specific variance, and all we need is this structuring matrix that predicts what the correlation between family members should look like if it's due to a locus in this region of linkage. And that structuring matrix is based on a concept called identity by descent. We want to know the proportion of alleles that a relative pair shares that derive from a common ancestral source. That's the key. I don't actually care whether they have an A allele or a T allele. I want to know whether they got the same thing from their grandfather or not, because I'm not actually assessing the effect of the genotype. I'm using the genotype to mark the movement of chromosomes through families. So all I really care about is, do they share or not share? And this matrix in front of our linkage component, our IBD matrix, is essentially for each pair of individuals half the probability that they share one allele identical by descent plus the probability that they share 2 alleles identical by descent. That's it. And the simple concept behind it is that in a region containing a locus that influences our traits, relatives who are phenotypically more similar will share more alleles identical by descent than relatives who are phenotypically dissimilar. If you think about it for a while and scribble on the back of the paper, that's true for pretty much any model, any genetic model you write down. If it's dominant, if it's recessive, if it has a small effect, if it has a large effect, if we're in the right region, relatives who are more similar should share more alleles IBD. The key to this is that it's kind of an improvement over our additive genetic kinship prediction. So when we're looking for additive genetic effects, we expect that relatives who are more closely related will be more correlated than relatives who are more distantly related. This is an attempt to improve upon that and say, okay, not only should siblings be more correlated than cousins, but siblings who share in this region of chromosome 12 should be more correlated with each other than siblings who don't share in this region of chromosome 12, essentially. So here I have some disease of interest. Yeah.
The two are not, you have to choose the two factors in the model, because I guess if you have the first one,
Yes.
you have basically not
Yeah, no. You always want to include that additive genetic component in there. That's an aggregate of effects all over the genome. And for any of the traits that we study here in this room, I'm willing to bet, we expect that they are complex and influenced by multiple genes and their interactions with each other and with the environment. So I don't want to set up a hypothesis where, so if I leave that out, that additive genetic component, just cross that out, imagine it's not there, then the models I'm comparing are allowing for a correlation among relatives via this IBD, or no correlation among relatives. But we're already studying this trait because we know it's correlated among relatives. So you want to carry this component along and say we know that relatives are correlated. We're trying to, out of that aggregate genetic effect, we're trying to pull specific loci. So it's an attempt to get specifics out of that aggregate, but we want to allow for that aggregate to still be there to account for genetic effects not due to this particular locus.
At the same time, the P matrix would be based on the correlation of the whole genome.
Yeah. Yes, absolutely. So you could, this is based just on the pedigree. It is possible, and some people do, estimate an empirical kinship matrix by looking at sharing over the whole genome based on markers they have genotyped. Yeah, in which case you may choose to substitute that. So I have a colleague who is studying host-parasite interactions for malaria, and using these sorts of models to look at that. And he has collected malaria, I don't know what they are, I don't work in that field, but he's got little malaria things and has genotyped his little malaria things, but of course he doesn't have a pedigree for his malaria. So he uses an empirical kinship matrix to account for overall similarity, genetic similarity among the parasites.
There was a paper earlier this year that looked at the relationship between empirically derived pedigrees versus typically derived pedigrees, and showed that while both could, in humans, and human pedigrees actually in the same sample, showed that while both could be quite predictive, typically the one that you're actually, the standard way of getting into based on support is typically strong, because there's some variation in the way you estimate the empirical pedigree.
So did everyone hear that? There's a paper out that in a situation in humans where you could have your choice of either has shown that you're better off using the pedigree-derived kinship, at least based on the information sources we have right now. Yes?
So what you see yourself as doing is pulling some variance out of this overall kinship measure, but do you want to do it in a sort of hierarchical approach where you say, let's explain everything we can by this first measure, but then keep the other one in as well subsequently? Or?
Yep, yeah. And you can easily do that. So you can, as usual, the solution is add a component. So you can say, I accept that ApoE is important for Alzheimer's disease, and I'm going to account for ApoE as my first QTL, but then continue and add a second QTL and look for that after taking into account what I know about the first one. Yep. And you can do the same sorts of things with association models or hybrids of association and linkage. So a postdoc working with me, Ellen Quillen, has just submitted a paper on paraoxinase, where there's a well-known major effect of the structural locus. And we are looking in a GWAS for additional possible loci after accounting for the whopping huge effect of the structural locus. So yeah, accounting for known loci can help you in the same ways that accounting for known covariates can. Yes?
After accounting for the known loci, does the heritability estimate drop?
It depends. It depends how you parameterize it. So you remember, I'm looking at it as a proportion of the total. If I'm taking that known locus out of my genetic component and I'm looking at the unexplained as a proportion of the total, it drops. If I'm taking that known locus out of both the numerator and the denominator, then not necessarily.
But as a covariate, it's only accounting for...
So our goal is that as a covariate, it's coming out of the denominator. Well, okay, unless we're doing association studies, in which case it's coming out of both.
Sorry, I meant heritability estimates.
Yeah, so I'm thinking about this situation here, where our heritability is the aggregate of the effects of the loci over either the total variance or the total unexplained variance. I can take a genetic effect, then when I put in BMI, you notice I've taken out G1, G2, and G5 effectively from both sides, both parts of my equation here. They're coming out of here because of BMI, and they're also coming out of here. But I can also then look at my genetic signal as a function of the original total variance rather than the residual variance after having corrected for my things. So you can typically the numbers I show you will be like this, but numbers other people show you may be of the original total phenotypic variance rather than the residual phenotypic variance.
So if I'm thinking about this, you could, for example, model, if you took an illness where there were a number of different risk variants that have been proposed, right? You could easily model the percentage of liability that those variants have independently and in aggregate.
Yes.
While continuing to look for additional variants.
Yes.
That's very cool.
Yeah, and you've reminded me that I don't have the slide in here. But I should say a word about liability. I don't have the slide in here, so imagine along with me. Imagine my bell curve world. When everything I've told you is about quantitative traits, you can use these sorts of methods to analyze dichotomous yes-no traits. And the way that it's done, at least in SOLAR, is to effectively take my little bell curve world and impose it onto your yes-no trait, and say that behind your little boxes of yes and no, there's really some unobserved quantitative process that we call liability. And once someone's liability is over a certain threshold, they become affected. And that threshold is placed so that a portion of my bell curve equal to the prevalence is over the threshold, essentially. That is the conceptual model that is used to analyze dichotomous yes-no traits in this variance component system. It is a sacrifice of power because all we can measure about someone is which side of the threshold they're on. We know whether they're affected or unaffected. We may know that the position of the threshold changes with age, prevalence increases with age. We may know that prevalence is different for males or females. So the covariates in this case move around the threshold. But for any, so to do the analysis, essentially we're integrating over the range of trait values someone could have given their age and sex and affection status. that's losing power. We may be in a better position if we can get a trait value we can actually measure that's strongly correlated with that unobserved underlying liability. So that's the hope with these sort of biomarker endophenotype approaches, that we're measuring things that we can get to quantitatively that are correlated with that unobserved liability, essentially. And that is why I would rather have BMI than obesity. Yeah, essentially it's the same thing. Yep. Okay, linkage, identity by descent. All right, so in the linkage analyses, I'm looking for a place where these two affected individuals share more alleles identical by descent than I would expect by chance. Except these two siblings, we already know from our kinship matrix, are expected to share half their genome by chance. So it's not a very rigorous prediction. If I've only got SIB pairs, I need a lot of them to get to a particular linkage region with any confidence. If I've got more complex family structures, the chances of all three of these siblings sharing the same thing from their father by chance is lower, and so forth, and so on. So for linkage, concentrating the same number of examined individuals in fewer pedigrees increases your power. Also, linkage is somewhat robust to allelic heterogeneity. What's allelic heterogeneity? So this is a genetics concept. We've got locus heterogeneity, we've got allelic heterogeneity. We all think that the things we're looking for are complex, they're influenced by multiple loci. Locus heterogeneity is a power problem for both linkage and association. If there's something on chromosome 2 and something on chromosome 6 and something on chromosome 7, the more of those somethings there are distributed across the genome that account for that overall aggregate genetic component, probably the more difficult it is going to be to find each one. Allelic heterogeneity is when we've got neuregulin, and it's one of our important players, but there's a different allele in neuregulin influencing the phenotype in each of our families. So let's say each of these pedigrees has an allele in neuregulin that is influencing disease risk, but they each have a different allele. If we're doing linkage, we're still okay, because we haven't predicted anything about specific alleles. We've just predicted that these relatives who are similar should share. And that's still true. And we can still get to that even if these two are sharing a different allele than those two are sharing. Our linkage test still works. Association does not work so well under allelic heterogeneity because we're testing each allele. And so we lose we lose a little power when there's a bunch of alleles in the same gene. We have to find each of them individually rather than being able to find each of them in aggregate. As I mentioned in my talk, the models we've been using to date for linkage analyses have effectively tried to impose the same effect size on each of those alleles. So even though we're measuring share, don't share, and it doesn't matter which allele they share, we're trying to impose a single effect size on all of those alleles. So it's possible that these pedigree-specific analyses can do better under conditions of allelic heterogeneity by allowing for heterogeneity in effect sizes, essentially. Association analyses are more powerful than linkage analyses when you've got that situation where you have a functional variant, it's in hand, and it's accounting for variation across pedigrees. So effectively, association analyses are exploiting the fact that you have some founder locus back here that has been transmitted to multiple families who are actually related in a way back sense, but whose relationships we do not currently know based on the data we have. Also, we're modeling linkage as an effect on the trait variance. We're modeling association as effect on the trait mean. We have more power for that to test that fixed effect on the trait mean. So if I had the functional variant in hand and I could test whether I could have my choice of a linkage test or an association test, I would pick an association test. Okay, so that is sort of the variance component worldview 101. My plan was for us to take a little break now, and then for me to tell you how you actually run these models in Solar to get your heritability, test your covariates, and run your association analysis, and then one slide about linkage. Does anyone have any additional questions? Yes?
So, based on what you were saying then, for a linkage study, if say, for example, you're looking at anxiety, presumably it would be better to have anxious cousins than anxious siblings in order to maximize your
it's a complex question. So purely from the point of view of the geneticist, if I could have sibling pairs to do my linkage screen with, or I could have cousin pairs, the same number of cousin pairs will probably give me more power because there is less genome sharing by chance in those pairs. But when I go to my clinician friends, they will tell me that it's much more difficult to get cousin pairs than sib pairs, and that the rate limiting, it may be that the rate limiting factor is not how much it costs to measure the phenotype, but the recruitment costs. And that, in fact, I could have twice as many sib pairs as cousin pairs. Or you're in a situation like autism, where effectively all the multiplex families in the country have been ascertained. And, and it's also, you don't have to, in some ways it's a false dichotomy. You don't have to choose. You can collect both those families and put them all in the same analysis.
I guess the interesting thing would be where, say, if you were looking at doing anything where it's very easy to screen a huge number of people where there's a fairly common phenotype, then that would say you should ideally pick the ones which would share least variance.
Yeah, and so, yes, essentially. And in the days where the genotyping was the most costly item, those are the sorts of designs that people were advocating, along with the kind of extreme discordant pairs as well.
The pedigree that you have more complex relationships in it, right? Just think about it. The more complex relationships, the more precise, the more specific your pedigree matrix is going to be.
Yeah, two siblings and a cousin is an even more specific prediction.
Fit that or not fit that, the more confidence you're going to be able to have in the statistic that you have, and the more specific I think you're going to be able to get the linkage. Right? So it's not, it shouldn't, it's more of an issue of making the families more complex themselves than it is of choosing what pair of relationship.
And that's a simplification, but I was just wondering. So again, you might say you'd rather have a first order and a second order cousin for the sibling, the first, like if in a world where you're constrained by how many people you can enter into the study, for example, then those are the kind of driving principles.
Yeah, I've said this before, but there is no one size fits all. So the answer to all of these questions differs by what phenotype you're studying. A lot of what I'm studying is normal variation. I can study cholesterol levels in random people off the street. So I can say my first priority is to get gigantic families and then get their cholesterol levels. But if I'm studying autism, my 1,000 people off the street aren't going to give me very much information necessarily. And if I'm studying Alzheimer's, it's fine to say I want affected sib pairs, but when you're working with an elderly population, that gets more and more difficult. And then saying affected cousins are even better, people throw up their hands. So there are the designs that are optimal from a genetics point of view have to intersect with the real world of what's available from a phenotypic point of view. How long a break do you guys want to take? 10 minutes? 5 minutes. All right, Peter and David say 5 minutes.